folks in the back, I hope you paid a little bit less than the folks in the front. All right, welcome. Today we have one of our departmental colleagues, Professor Ernest Frankel, presenting our seminar. Ernest is well known to many of you. I'll just give you a little bit of his background. A very interesting trifecta with respect to his educational degrees. First of all, his bachelor's from Harvard in both chemistry and physics, and then his PhD to finish that triad in biology here at MIT, working with Carl Pavo for his PhD thesis. Postdoc at Harvard Medical School, where he was quickly snapped up by the Whitehead Institute to be a Whitehead Institute Fellow. And that was where I first met him, when he was the Whitehead Fellow here. And his background has moved from structural biology mainly now to the realm of systems and computational biology, combining molecular properties with complex networks. You'll see that here. Combining omic experimental measurements with computational network modeling. His work has been recognized by the Merrimack Prize for Systems Biology, and he's also an associate editor of the Journal of PLOS Computational Biology, which is the leading journal in our computational biology field. Many of you know he's been an excellent contributor to our undergraduate programs and our graduate program. He teaches at 2320, analysis of biomolecular and cellular systems, and co-teaching with the biology department. Our numbers are 2390 and 490, Foundations of Computational Systems Biology. He's also been very active in our departmental campus service, especially education, associate chair of our undergraduate program committee, and chair of our departmental computing facilities committee, and a member of the school-wide committee on school engineering, committee on educational programs. So, but we're here to hear about his research, and I'm delighted to turn it over to Ernest for that.
Thank you very much, Doug, for that kind introduction. And I also want to thank all of you who came out. I realize a lot of your plans probably got disrupted because of the hurricane, so I appreciate seeing so many of you here. So my work focuses on trying to find out the mechanisms of disease using new engineering-based approaches. And before I tell you about our research, I just want to provide a little bit of historical context. This plot shows rates of death in the United States over the 20th century. And you can see it's dominated by a couple of important features. The one that's least relevant to my talk here, but probably the most striking, is this blip in 1918, which many of you probably realize is the Spanish flu of that year. But what's more relevant for this talk is the sharp rate of decline in death in the United States in the first half of the 20th century, and then how that relatively flattens out towards the second half of the century. And the effects are more dramatic if you compare infectious disease and non-infectious disease. So at the beginning of the 20th century, infectious disease accounted for roughly half of all the deaths in the United States. And by the end of the 20th century, it was a very, very small fraction. So what accounts for this rapid decline in deaths from infectious disease? Well, the first part of the century was really dominated by engineering approaches to how to live in cities and how to develop safe foods. Work that was done in many places on sanitary engineering and food science, but including here at MIT, work by people like Ellen Swallow-Richards, Sedgwick, Underwood, Prescott, some of the ancestors of our department, really solved many of the human health problems of the early 20th century. And then in mid-century, the pharmaceutical industry takes over with the development of antibiotics that really causes almost elimination of infectious disease as a serious cause of death in the United States. Obviously, it remains a cause of death in the rest of the world. And so at the end of the 20th century, you're much more likely to die from heart disease, pulmonary disease, stroke, than you are from infectious disease in the US. So this red curve is a very encouraging one. The blue one is not so encouraging, right? We really haven't made much progress by this metric in curing non-infectious disease. So how did the pharmaceutical industry do such a good job here, and why is it not doing such a good job over here? Well, in the early parts of the 20th century, a lot of pharmaceutical development was dominated by the kind of approach that you're probably familiar with from penicillin, or I'll give a slightly more relevant example to our research, metformin. This is, to this day, the most commonly prescribed drug for type 2 diabetes. It was originally marketed in the 1950s. Its origins date back to herbal remedies from the Middle Ages. And it was many, many decades after it was introduced commercially that anyone was able to understand in molecular detail how it works. So it's possible, and was very successful, to develop drugs without any knowledge of molecular mechanism. The downside, though, is you have to do a lot of experimentation on animals and people at a level that's probably no longer considered ethically justifiable. And so the switch went from this kind of discovery for drugs, which is agnostic to molecular mechanism, to one that's entirely driven by molecular mechanism. By understanding the molecular biology of a problem, you can then try to find key proteins to target that'll block the disease. And a very successful example of that is the work on HIV. So this cartoon indicates the HIV viral life cycle of the virus that causes AIDS, how it enters the cell, replicates, and eventually leaves. And those things which are outlined in red are places where the pharmaceutical industry has developed chemicals that specifically target individual proteins, and those are able to block the disease. And through these individually, now more powerfully in combination, it's possible to take something that was a death sentence just a few years ago and make it a chronic disease, but a manageable one. So to what extent does this model of understanding the molecular biology of a disease and then making these magic bullet drugs that target exactly one protein work? Well, it's worked very well for some diseases, but increasingly we're seeing many, many, many failures. So here are some examples of drugs that have failed just in the last few years at a very late stage in the drug discovery process. All of these had extensive validation that the targets made sense from a molecular biology standpoint. They were tested in vitro in animal models for safety, and then most of them failed at a very late stage, what's called phase 3, when they're actually gone to see whether they actually work in patients. And statistical studies have shown that in about a third of the cases when things fail at late stage, they fail because they're simply not effective. So there's been a breakdown in this idea that you can get the molecular biology view of the world, find the protein that you need to target that protein and cure the disease. That's not going to work anymore. And so our approach is really to try something new, which is to use systems biology-based approaches, to really comprehensively, systematically measure at a molecular level what's going on inside cells, and then use that systematic knowledge together with computational models to try to identify places where you need to intervene. And it's probably going to be usually plural, not just one place where you need to target, but multiple places. So what I'm going to do in the coming talk is tell you about the kinds of data that we've been using, the kinds of models we've been using at a high level, and then take you through some specific examples. Many of you are probably familiar with the first comprehensive assay to be developed for a class of molecules in eukaryotic cells, the microarray, which allows us to systematically measure gene expression levels across the organism, and now that's rapidly being replaced by sequencing-based approaches. So that's comprehensive. It tells you about almost every mRNA in the cell. Now, mRNAs are not in and of themselves particularly interesting, but they give us a very precise molecular readout. There are other molecular readouts, for example, protein-DNA interactions, protein-protein interactions, post-translational modifications on proteins, levels of metabolites that we can measure systematically but not comprehensively. And then there are other assays based on screens, genetic screens, to individually knock out or knock down individual genes, or use chemical compounds and measure phenotypes. So all of these technologies usually go by the name omics for proteomics, genomics, transcriptomics, metabolomics, and I may refer to them that way. So when we approach these omic data, we're trying to take all of these data and develop a model of what's going on in the cell. What are some of the challenges that we face? Well, one of the first challenges that people noticed when they started using these omic data on systems where they knew what was going on was that when you systematically measure what's going on, you find out that the expected response is a tiny fraction of what really happens in the cell. And I think this was first identified by Audrey Gash in yeast, but it's been seen now in many, many systems. These are data I'll refer to later from the White Lab, in which they looked at mutations in the EGF receptor. The EGF receptor has a downstream signaling pathway that's been extensively studied, but only about 10% of the phosphorylation events that occur when you mutate the EGF receptor actually occur in the pathway that you expected. And a big chunk of the changes occur in pathways that are unannotated. So this is a bit disturbing in the sense that you can't simply take these data and paint them onto known molecular pathways and read out automatically what's going on in your cell. But on the other hand, there's a big opportunity here, because all of the molecular biology-based approaches have been focusing on this 10% of the pathway that's known. So it could be that in the remaining 90% of the data, we have the opportunities for new therapeutic approaches. So this is the first problem that we notice when we look at omic data. But a second and perhaps more disturbing problem is what happens when you apply more than one technology to study a biological problem. And this was first pointed out to me by Leona Sampson, and we've since seen it in many, many systems. So if you take a simple system or a more complicated one, and you do a microarray to measure which genes are changing in expression, and you simultaneously measure which genes, when knocked out, affect the phenotype, you might expect a certain degree of overlap among the genes, or certainly among the categories of genes that you find. But in general, what you get is a Venn diagram that looks more like this, that there's no significant overlap between the hits from different kinds of technologies, microarrays, proteomics, genetic screens, what have you. Now, the pessimist might say that that's simply because each of these assays is extremely noisy. And if you have very noisy assays, then you can't expect much overlap. But we know that's not the case because individually, these assays are extremely reproducible. So it must be that there's some sort of systematic bias that's causing each assay to find out something different about the cell. Again, this is a problem if you had hoped to simply take the overlap and use that as the high confidence hits from your experiments. But it's an opportunity because each of these is telling you something new about the cell if we can just figure out what it's telling you. So two postdocs at the time in my lab, Esti Jaeger-Lodem and Laura Riva, tried to tackle this problem. They now each have their own research groups in their home countries. And what they did was they looked whether there was any systematic bias between the kinds of genes and proteins that you get from different assays. And what they found is, in fact, that is the case. So in genetic screens in yeast, you tend to get master regulators of responses. You get transcription factors, kinases, phosphatases as coming out here, but you don't detect those as differentially expressed statistically, although in individual cases you certainly do. And similarly, there's a bias in the expression data, which tends to find effector molecules, metabolic enzymes, and the like, and those don't tend to show up as often in the genetic data. So to make this concrete, let's look at DNA damage. And I've just shown you a small fraction of all the data from DNA damage, but some of the things that come out as genetic hits are these proteins, the sliding clamp checkpoint, that runs along the surface of the DNA and detects places where there are DNA damage events. And then initiates a signal transduction cascade that includes proteins that were also picked up in the genetic screen. And that eventually transmits information to the nucleus, causing cell cycle arrest and DNA damage repair. So these things show up in the genetic data. They don't show up as differentially expressed. Things like ribonuclease reductase, these repair enzymes, show up as differentially expressed, but not in the genetic data. So why is that? Well, think about this for a second. These proteins are detecting the DNA damage. They have to be on all the time in the same way that the smoke detectors in your house have to be on all the time. You can't wait for the fire to turn them on. You can't wait for the DNA damage to turn on the things that's going to detect DNA damage. However, the repair enzymes, you have an advantage to leaving them off in the same way that we keep, hopefully there are some sprinklers in here, we keep the sprinklers off all the time, and we only turn them on when there's an emergency. So the cell is doing the same thing. It has the detectors on all the time. If you knock those out, you don't detect the DNA damage. It has a huge effect in phenotype. But it turns on the repair enzymes, the effector molecules, only when necessary. And so this systematic bias actually provides us with an opportunity, because we can see that there, in this case, is a pathway that goes from the master regulators identified by the genetic hits down to the effector molecules detected by the genetic expression, by the expression data. And so if we can reconstruct this pathway, we might be able to find paths from the data here to the data here. And along the way, we might pick up some of these molecules that are well-characterized pieces of the DNA damage response, but that were not detected in any of these assays. So this is our overall goal, and I'll just show you on a cartoon then. Imagine we've got some cell process that starts with an extracellular receptor, eventually having both proteomic effects and effects on the nucleus. And I've colored with different colors hits from mass spectrometry, genetic hits, and differential expression, and intentionally left them, every protein only gets one of those colors. There aren't very many things that are going to be detected simultaneously as both genetic hits and differentially expressed and mass spec data. But there may be pathways that contain some of each of those. And so one of our goals in this approach is to try to find these pathways, which may never have been called a pathway before, but are indicated partially by each of these different types of data sources. And the second goal is to try to find some of the proteins that were missed by all the assays, the ones indicated in red, that could be just as good for drug targets as the ones that were directly detected. And so we're trying to shift how people view omic data from just mapping to known pathways or looking for overlap among disparate data sets to a more integrated view that tries to find a physical link among all these proteins and genes that are identified by the different assays. So what I'll show you today is that we found ways to actually find these links among proteins, to find network models that incorporate data from the many different assays. But we need to be careful in how we do this, because if we take naive approaches, we rapidly end up with these what we call lovingly hairball networks. that don't convey any useful information. They don't guide experiments. We want networks that actually help us design new experiments that can get us closer to therapeutics. So two of the lessons we've learned from these approaches is that while network models can help, not all network models can help, and I'll tell you how we've solved these problems. Now, the third important point I want to make before I delve into some of the details is what we do with all the expression data. It's obviously relatively inexpensive right now to map mRNA expression changes in cells. There's a lot of it already in the databases, and it's pretty natural to assume that that's going to be some sort of noisy readout as to what goes on at the level of proteins. And so if I can take the expression changes and map them to proteins, I can figure out what proteins are changing. But it turns out that's rarely the case. So several studies have shown that if you compare expression levels and protein levels, there is a crude correlation. In this study, it was an R-squared of 0.2. But it's only a statistical correlation. And at any value of the mRNA expression level, there can be 3 orders of magnitude difference in the protein levels. And so if I use mRNA simply as a proxy for proteins, I'm going to be going wrong most of the time. So our approach instead is to use mRNA levels as evidence of upstream signaling pathways. And we do that in just 2 steps. First, we go from the mRNA data to figuring out what DNA binding proteins were binding to the genome and changing the regulation of those genes. And I'll show you how we do that using sequence data and using experiments. And then from there, we can build up to connect to other kinds of proteomic data, mass spec data, genetic data as well. And this we'll do using protein-protein interactions. So this is our overall scheme. And as I said, it requires us to get good at 2 things. One is being able to map transcriptional regulation, and 2, coming out with network models that make sense of these data in a way that guides experiments. And so in the rest of the talk, I'll take you first through our work on mapping transcriptional regulation with just a few quick vignettes on problems that we've been studying, and then go into much more detail on some of these network modeling approaches. Now, my first foray into this area of omics, as Doug mentioned, was when I was a fellow at the White Institute, and I teamed up with Rick Young, and together we developed a map of the transcriptional regulatory binding sites across the genome of yeast. This is a small piece of that map. The gray boxes indicate genes. The colored boxes indicate binding sites for transcription factors. And using both computation and experiment, we mapped out the binding sites for almost every transcription factor in yeast across the genome. It was the first time that had been done for eukaryote. And even though this is now a relatively old publication, it's 8 years old, it remains the standard in the field. It's still used very widely. It's been cited over 1,000 times. Now, it spawned a lot of interest in mapping out regulatory networks, binding sites in many other genomes. And the immediate question came up, can you do something similar in yeast that you did in yeast in mice? Can you do something similar in humans? There are a lot of problems. I mean, most obviously, yeast have one cell type mostly, whereas humans have hundreds of cell types. We have lots of organs we need to consider. But there are also genomic issues to consider. There are an order of magnitude more transcription factors and 3 orders of magnitude more DNA. And so the experimental methods that worked so well in yeast do not work particularly well in humans and in mice. So another approach that people thought about at this time, as the human genome sequencing was ending, was, well, we have all this surplus sequencing capacity. What if we sequence lots and lots of species, which they went ahead and did. And so here's an example of a tree showing some of the sequenced species. This is just vertebrates. We have everything from alpaca to zebrafish having been sequenced. Now let's line up all those genomic sequences and see what we find. So there'll be some regions where you get strong conservation because you've got protein coding genes that are conserved across evolution. And then you'll have some regions which are very poorly conserved. These might be what people used to call junk DNA, although they seem to be regretting that term. And then there are some regions where there are very short sequences of conservation indicated by this box. And those are going to be likely to be binding sites for transcription factors. So what about just doing lots and lots of sequencing, which could be relatively efficient because we have all the infrastructure in place, and finding these regulatory maps purely on an evolutionary basis. So while all this was going on, no one bothered to ask the question whether binding sites should be conserved in evolution. Do we have any experimental data to indicate that binding sites are conserved during evolution? So I teamed up with David Gifford in ECS, just a few floors upstairs, to directly answer that question. We worked together with 2 very talented postdocs, Robin Dowell and Duncan Odom, who now have labs of their own. And we asked the following question. If we look at highly conserved transcription factors, which play major roles in the liver, and we look at those transcription factors in human liver and in mouse liver, what do we see? Do we see that they're binding to the same sites, or do they differ? And our results really shocked people, because what we found was that it was a very, very small fraction of all the genes that we looked at that had exactly the same binding events in both human and mouse liver. even though the proteins that are binding are highly conserved. In the vast majority of cases, things were changing around dramatically. So this means that you can't simply rely on sequence conservation. There will be conserved binding events, but there'll be a lot of non-conserved binding events. So experimental techniques are going to be critical in understanding the regulatory networks in humans and in mice. Now, another thing that's a bit odd about this is if you have so much change in the regulation, how can you have highly conserved transcription? And it turns out that liver transcription is very conserved between humans and mice. And so we started to do some computational modeling to try to address this question. And Kenzie McIsaac and Alice Lowe, who were grad students in the lab at the time, worked on this. Kenzie's now a senior researcher at Merck, and Alice is a postdoc in Singapore, her home country. And what they did was they looked just within a species, they looked in mice, and they compared expression in the liver and the brain, and asked whether they could come up with computational models driven by experimental data that could explain differences in expression. And if you have efficient computational models, then you can ask some very direct questions. Do you need perfect conservation of binding sites, which is what you would also need to detect them in evolution? Or are there other ways to conserve gene expression without exact conservation of the binding events? And what they found was actually a relatively simple answer, that of all the models they considered, they could explain a very, very large fraction of the gene expression with an R correlation coefficient of 0.65 for gene expression changes between liver and brain by looking at where just one protein is recruited to the DNA. This is a protein that doesn't directly bind to DNA. It's indirectly recruited. But when that protein is recruited very close to the transcription start site, you have a higher degree of expression. And when it's further away from the transcription start site, you have less expression. And it did not matter what the proteins were that were recruiting it. So you can imagine during evolution then, as long as you manage to recruit these regulatory complexes to roughly the same site along the DNA, it doesn't matter if you have exactly the same transcription factors, and then you'll get a sequence conservation, or you have slightly different transcription factors that have moved 100 bases over this way or that way, and then you won't have the sequence conservation. So these computational models help us understand some of the mysteries that emerge from our study of the evolution. Now, we've been focusing more and more on less basic science and more medically relevant problems, and so I'll just tell you a few of the problems that we're working on at the level of transcriptional regulation. So we've had a very fruitful collaboration with Dr. Alan Cantor of Harvard Medical School, looking at some of the transcriptional regulators involved in hematopoiesis, the development of blood cells. We've mapped out the binding sites for the master regulator of hematopoiesis and some of the regulatory complexes that interact with it. This work was done by Lau Riva, who I already mentioned, and Tali Mazur, who was a technician in the lab at the time and is now a grad student at Stanford, and is continued now by Gabriella, who's a BE student. Another major project in the lab is looking at insulin resistance, and we have a collaborative project with Doug and Forrest and Roger Davis of UMass Worcester, in which we're looking at mice that have been fed either normal diets or high-fat diets. And then we're examining through a number of omic technologies, some done in our lab, some in Forrest's lab, what changes in these livers that make the high-fat diet mice resistant to the insulin response, resistant to insulin, whereas the normal diet mice are responsive. And that's right now at the stage of data collection, and we're eventually going to do a network modeling of that as well. And another major focus in the lab is on the study of Huntington's disease. Huntington's disease is a fatal neurodegenerative disorder. It's a dominant genetic disease, and it was the first genetic disease to be mapped to chromosomal location. And that work was done by an international team that was led by David Hausman in the biology department in the Koch Institute, together with, among many other people, Leslie Thompson. And we collaborate both with David and with Leslie. Leslie's at UC Irvine. And we've been approaching the study of Huntington's disease at a number of different levels. Most of our work so far has been at the level of transcriptional regulation. So we've done some hypothesis-driven research. We had reason to believe that the heat shock factor, HSF1, was driving some of the differences in response in these cells. And we examined the binding of this protein in cells that had wild-type and mutant Huntington. And we found that Huntington blocks some of the heat shock response, the stress responses. We've also looked at a number of different kinds of epigenetic modifications. And as a link to some of the network modeling approaches, I just want to tell you a little bit about some of the work that we've done building off of our observation done by Chris Ng in the lab, that Huntington induces relatively significant changes in DNA methylation, an epigenetic mark. So Chris measured, with help from others in the lab, DNA methylation in cells that had either wild-type or mutant Huntington. And he found that there were specific sites at which the presence of Huntington could either directly or indirectly, we didn't know at that point, induce changes in DNA methylation. And so he hypothesized there were DNA binding proteins that were influencing whether the DNA was methylated or not, and those DNA binding proteins would be present in only one cell type and not in the other. Now, if that's the case, you should be able to look at these sites where there are changes in methylation and use sequence analysis to try to figure out what the proteins were that bound there. And so, using a sophisticated computational approach, he identified these sequence regions that are associated with changes in methylation. He came up with a set of hypotheses that were validated by a number of different sequence analysis methods, and he found 4 families of transcription factors that he believed were likely to be influencing or influenced by DNA methylation. And then we carried out experiments to actually determine whether that's in fact the case. So we immunoprecipitated members of those families, sequenced the DNA, mapped it back to the chromosomal positions, and then we could show with statistical significance that sites where these proteins were bound more in one cell type than the other had less DNA methylation. So there seems to be a functional link between the binding of these proteins and the changes in methylation. So this is an example of the kind of work that we do to take transcriptional regulation and use it to identify the regulators, changes in transcription, epigenetic modifications, use it to identify these regulators. And then we're going to use that data to build up network models. So let me tell you a little bit about how these network models are made. We've approached this in a number of different ways. I won't have time to describe all of the approaches. I'll just mention briefly work of Sarah Goslin, who's developed a network modeling approach that's very specific for a certain kind of problem where we know that there are going to be multiple stimuli that induce similar responses, overlapping responses, but not identical responses. And an example of that she worked on was the epithelial mesenchymal transformation, which is a normal part of development, but also occurs in cancer. And in vitro, you can induce it with a number of different stimuli. And so it's likely that all these different kinds of EMT have some things in common, but some things that are distinct. And her network modeling approach, which is called multi-commodity flow, allows her to simultaneously identify those common pathways and the ones that are distinct. But I'm going to spend most of my time talking about work that was done by Carol Huang in the lab with help of others in the lab. She's now was a grad student at the time. She's now a postdoc at the Scripps. And she was studying glioblastoma. This is a brain tumor that is really a shockingly devastating tumor. It's usually identified at a relatively late stage. Patients come in with nonspecific symptoms. On scan, a mass is detected. It's surgically resected. They're treated with chemotherapy, but it invariably recurs. And most patients don't survive much more than a year after initial detection. Some of you may be aware that this is what killed our former Senator Ted Kennedy. So this is the kind of disease where we hope that new approaches might come up with insights that have been missed by the molecular biology approaches that have preceded us. We started with a very focused question about variant 3 of the EGF receptor. This is a specific mutation that occurs very commonly in these tumors, and higher levels of expression of this V3 mutant are associated with the worst prognoses. And we started with data that Forrest White and his lab had collected on cells, a cell culture model that expressed various levels of this V3 mutant, or an alternative mutant of the EGF receptor that's unable to carry out tyrosine phosphorylation. And so using a mass spec-based approach, they were able to provide us with very quantitative measurements of which peptides are changing in phosphorylation in cells that have the V3 mutant versus the control. So we took their phosphoproteomic data, we collected expression data, and we also collected one of these epigenetic marks that allows us to identify sites of regulation. And Carol very carefully did experiments of a type that I won't have a chance to explain, but it's another one of these epigenetic marks called DNase-seq, or DNase hypersensitivity, that allows us to find sites where transcription factors are binding without needing to know in advance what those transcription factors are. And then she developed a computational pipeline that looked in those regions at differential DNase-seq data, identified sequence motifs represented here by these little boxes, and asked which of these are most likely to be functional by asking whether they were associated with changes in gene expression. And so she goes from a very large number of potential transcription factors down to a shorter list, but importantly a ranked list that associates with each transcription factor probability that it's relevant to our problem by integrating epigenetic data and transcriptional data. So now she has all the pieces that we need to try to build these models. She has the phosphoproteomic data from the White Lab, expression data, protein-DNA interaction data that she developed through her computational modeling. The last thing we need is protein-protein interaction data, and there's a lot of this in the literature. There are 250,000, roughly, protein-protein interactions that are in databases right now. And these databases are really fantastic in that they allow us not only to know that a protein-protein interaction has been reported, but to trace back what kind of data underlie it. And so we can look at these data, sometimes it's called the interactome, maps of physical interactions. And so with all these data sources in place, we can then ask the question whether when we take the hits from the proteomic data, her predicted transcription factors, and the interactome, what do we get? And what we get, unfortunately, is a hairball diagram, which contains, as I said, just a lot of too many connections to be able to design any experiments based on it. And the reason we get so many connections is because even the best data set, apologies to Forrest if he's here, even the best data set is going to have false positives in it. And certainly that's true of the interactome, which has a lot of both false positives and false negatives. And when you think about trying to find connections among all these data, if you have wrong connections or if you have missing connections, all of those will lead to making this hairball larger and larger. And so in order to model this in a way that leads to experiment, we needed to take into account the fact that not all the data were real. There were false positives in the data. There were missing interactions. But also importantly, that not all those protein-protein interactions that are reported in these databases, this interactome data, are equally reliable. Now, this problem, fortunately, has been solved by others. There are very nice Bayesian statistical methods that allow us to, in an automatic way, to associate with every one of those 250,000 protein-protein interactions a probability that's true based on the kinds of data that underlie it. So you can go through the database in an automatic fashion, find out for that interaction, why do we believe that interacts? Was it because there was a yeast-2 hybrid experiment? Was there a co-crystal structure? Was there some combination of different kinds of evidence? And then based on what the evidence are, assign to this interaction a probability. So now we take that interactome. It has on every edge in an automatic fashion a probability. Now we just need to decide what to do with it. How do we find the pieces of that probabilistic interactome that are going to contain the most likely functional networks? So here, there's important intuition that we want to use, which is that in these little networks, the nodes that are indicated in this mustard color are proteins that we say are changing in phosphorylation, for example. And we want to ask, what network should we choose to connect protein A to protein B? Well, we'd naturally assume that the network on the left, the path on the left, would be more likely to be the real one because it picks up an additional protein that's changing in phosphorylation. Whereas the pathway on the right, we don't know anything about these proteins, whether they're changing in our condition or not. And similarly, when we try to explain the transcription factors that are explaining expression changes, if we're trying to figure out whether C or D is a better explanation for the data, we'd have more confidence in D because it's connected to a protein that was directly experimentally detected as changing in the experiments. So in short, we're going to prefer pathways that pick up more of our experimental data. And there's a way to formalize that. It's a thing called a prize-collecting Steiner tree. It's a mouthful. I'll explain exactly what that means. So imagine this is a little piece of our hairball network. And again, the proteins with this mustard color are the ones for which we have experimental data. The genes that are changing in transcription are the boxes at the bottom. And we want to go through this hairball and try to find pieces of the network, now indicated in purple, that we believe are most likely to be relevant. And so we want to ask, if we have a protein out here that was detected as changing, is it a false positive, or should I include it? Do I have confidence that this is part of the response? And the way we do that is, again, by this prize-collecting Steiner tree algorithm. So what are the prizes? Well, we tell the algorithm that it's going to get a prize every time it includes a node in the network for which there's direct experimental data. And the prize goes up the more confident we are that it's really changing. So the algorithm, if it comes up with this network indicated by the purple lines, gets a prize for this data point because it's included in the network, and it gets no prize for this one because it was excluded. It also gets no prize for the gray nodes because we have no direct experimental data to tell us whether they're relevant or not. So we ask the algorithm to try to collect as many prizes as it can. Okay? Now, if that's all you ask, you get that hairball diagram. So we balance that objective with one other term in this function, which is a cost. Every time it adds a new node, it has to add some edges to connect. And it's going to pay a cost for those edges. And it's going to pay more if it's an unreliable protein-protein interaction and less if it's a reliable one. So the net result is if you optimize this function, then the algorithm naturally has to decide, is it worth including this node? If this is a very high confidence proteomic data point, and these edges are high confidence protein-protein interactions, then of course they'll get included. If neither of those is true, then it'll get excluded. And the relative weights of the prizes and the cost will determine for the intermediate cases whether it gets included. So that allows us to go from these hairball diagrams, where there are potentially thousands of proteins in the hairball, to very small networks that directly lead to experiments. And so this is the network that Carol got when she combined the proteomic data, expression data, and epigenetic data. The prizes, the nodes with prizes are the ones that are outlined in red, and the strength of the prize is indicated by the color. There are also nodes in here that have no color, and those are ones that the algorithm is including because it says they're needed to explain the connections among the data. And I've highlighted here some pathways that make a lot of sense. So we see PI3 kinase, we see the MAPK cascade. Some things that probably make sense, synaptic transmission. It is, after all, a brain tumor. But staring at these networks is not going to be enough. We need to do some statistical tests to figure out whether, in fact, these networks are reliable. And so Carol, with help from others in the lab, did a number of statistical tests. She asked, for example, whether the nodes that are detected in these networks have been previously reported to be associated with glioblastoma. And there's a systematic way that you can do that based on a thing called GBM ranker, and she found statistical significance for that. She asked whether the network also is compatible with newer data sets that the White Lab just published a few weeks ago. And in fact, that is true. So these networks aren't just explaining what's going on in cell culture, but they're actually explaining what's going on in some xenograph data that we'll talk about in a minute. And lastly, with help from others in the lab, she was able to show that the transcription factors that she identified in her networks actually have some explanatory power in looking at patient data, which was actually quite surprising. So by looking at the TCGA, the Cancer Genome Atlas data, you can find expression data for patients who have the same V3 mutation and ones have other genetic abnormalities. And the transcription factors that we find can be associated with statistical significance with the genes that are changing between the patients with V3 mutants and the ones that don't. So these were statistical tests, but we also wanted to do some direct experimental tests to see whether these networks actually make sense. And one thing we realized we needed to test were whether these proteins that are reported only by the network, included only because of the network optimization, whether there's any experimental support for them. So here's one protein, P300. It's a protein that's known to have many, many interaction partners. It's a very sticky protein. It's what's called a hub in this field. And you might imagine that the reason the algorithm is sticking this in is because it just needed to find some way to connect up everything. And so it used this protein, and it would have put this in regardless of what kind of data we gave it. So to experimentally test that, we asked whether it really is associated with the sites that the algorithm said it was. P300 is a protein that's a transcriptional regulator, but it doesn't directly bind DNA. It's recruited by DNA binding proteins. The algorithm is saying it's associated with particular transcription factors. So we can immunoprecipitate this protein, measure the sites to which it's recruited, and then ask whether those are in fact sites of transcriptional regulation. So we compared the sites that were bound by this P300 protein that the network said was relevant to the sites that were actually changing in this DNA-seq data that I mentioned previously, the sites where we know some transcription factors bound. We say a strong statistical significance between P300 binding and sites that are changing in hypersensitivity between the mutant cells and the control cells. And so this is a pretty strong indication that P300 has some specific role in transcriptional regulation in these V3 mutants compared to just some sort of generic role in transcriptional regulation overall. And when we look at the types of genes that are bound, we find that these also make sense. So we find genes involved in cell adhesion and development, both of which, in terms of the biology of glioblastoma, do make a certain amount of sense. So these were statistical tests of our network, experimental tests of individual proteins. But what we really wanted to get at was whether this helps us identify therapeutic targets. And so what Carol did was she ranked every protein in the interactome by whether it was in her small network or very closely connected to that small network. And those, she would say, would be very relevant to the disease process, or whether it was far removed, which would be less likely to be influencing this disease process. And then she went out, looked in literature for chemical compounds that specifically interacted with the proteins that she selected, and did the experiments herself to determine whether targeting these proteins could influence the growth of these tumor cells. So on the top are the nodes, the proteins that are most closely associated with her network. And on the bottom are proteins that were less closely associated with her network. And she measured at a relatively constant dose of drugs to target these proteins whether it blocks growth of the cell. And you can see that in most of the cases for the best targets, you get significant reduction in the growth of these cells compared to the lower ranked targets, which are not particularly effective at these doses. Some of these nodes are actually rather surprising. For example, the algorithm said that the estrogen receptor would be relevant to glioblastoma. That's not something one would have expected based on the literature. In fact, I was rather disappointed that Carol was going to go and test this, but she went ahead and did it. She tested whether tamoxifen blocks the growth of these tumor cells. In fact, as you can see here, it's one of the best hits that we got. So we get very non-intuitive results from this unbiased approach, letting the data guide where we're going to go rather than using our own preconceived notions about what's relevant. And you can also look at whether there's differential killing of the tumor cells that have the V3 mutation compared to control cells that are also glioblastoma cells but have an inactive kinase. And we can find is for the best targets, we get more often a statistically significant difference in the killing of the tumor cells that have the V3 mutation versus the controls. And that's much less likely to occur, and the magnitude of the differences are much, much smaller for the control targets. And finally, in this context, we were able to find that there were certain combinations of these drugs that were actually somewhat synergistic. And so this suggests we can take a network model based on phosphoproteomic data, epigenomic data, and expression data, and use it to guide experiments. We can go from having a list of 4,500 proteins that we want to consider to a list of 30, 40 proteins that we want to consider, and then do the experiments that will lead us to compounds that can actually block the growth of these cells. Now, more recently, Nurjan Tujbag's lab has been trying to develop more sophisticated versions of both the algorithm and applying it to more sophisticated models of glioblastoma. And I'll tell you briefly in the time that remains about her work. So in Carol's approach, we were using something called a prize-collecting Steiner tree. And that takes some complicated network and defines what's called a tree structure. And the important piece of what a tree means in this context is just that there's a connected component. Everything is connected to each other. There's one path, there's one network that explains everything. Now imagine that this is the true network in these cells that's altered in glioblastoma. But for whatever reason in our experimental data, we're missing the key connection between those two pieces of the network. So what's the algorithm going to do? Well, it has basically two choices. It can tell us half the story. You can find which either whatever half of the network gets a better score and just tell us this, or it can find some spurious way of connecting these things up and then report the whole thing. Neither of these is a desirable result. What we'd really like to do is get back two different networks, and the algorithm say, well, I don't know what the connection is between these two processes, but they're both present in the data. And so that's what's called a Steiner forest approach, a forest being made up of at least two trees. So we get these two or more trees in this result. And the more realistic data that we're working on, we're now moved from a cell culture model to xenograft data. And this, again, is work from the White Lab that we've been analyzing, work specifically of Hannah Johnston. And what she has is tumors from 8 different patients with diverse genotypes that have been propagated in mouse models, and from which they've collected very carefully mass spec data on phosphoproteomics and global proteomics. So now we can take this prize-collecting Steiner forest approach and apply it to each of these patients and see what we get. Here's an example of one of those patients in which we have a forest of trees in which every tree is rooted in a cell surface receptor. And then it's been clustered based on just the connections among these proteins. And so some of the pathways that emerge, we see over here, number 1 is the TOR signaling pathway. Over here is a glutamate response pathway. Some of these pathways that seem to make a lot of sense. Now, another one of the things that Nurjan has been working on with others in the lab is how we can assign statistical significance to these networks. I showed you previously that we were worried about this protein P300. Maybe it was in the network simply because it's a very sticky protein and interacts with a lot of other proteins. Is there some way, without having to do experiments in each and every one, we can determine whether a node is here because it's really relevant to the data for this particular patient, or whether it's just there because the network had to find some way to connect things? And so we've developed a randomization approach where we run random data through exactly the same pipeline, and then we ask how often does each protein show up in these randomized networks. And now we can take those previous networks and assign to every node in the network a probability. And the more red it is, the more likely it is to be specific to this patient. So you can see that the TOR signaling pathway, this glutamate pathway, they show up as all red. They're very likely to be specific to our patient and not just there for some network property. They're individual proteins, GRAB2, for example, the EGF receptor itself, which would have shown up pretty much with any data that we gave the algorithm. And so now we can take that final step and go from these network models to try to predict therapeutic targets that would be common for all patients, and ones that would be specific to individual patients. So this shows some very preliminary work where we've looked at the 8 different xenograft models for these 8 different patients. And in blue is a case where there's no, the networks the networks say that a certain protein is not at all relevant, so each column is a different protein. And in red are these probabilities. So you can say there are certain proteins, indicated up here, which are common to all or most of the patients. And we would argue that these are targets that might be likely to be effective for all of these patients. whereas there are other targets that are specific to some subset of the patients. And so perhaps NF-kappa B signaling, compounds that target NF-kappa B signaling could be effective for some of these patients, whereas compounds that work on the TOR pathway would be relevant for other patients. So what I hope I've convinced you is that we can take these many different kinds of data, each of which tells a different complementary story about what's going on in the cell, and use these systematic measurements together with computational models to drive experiments that can guide us to compounds that can have an effect in vivo in a much more effective and unbiased way than either a traditional molecular biology approach or a naive approach. Now, the real test will come with how broadly we can apply these methods. And so we have a large number of projects underway to test this in many different systems. Some of these are being done largely in our lab, some with collaborators, many projects on cancer and neurobiology, and then a range of other things, ranging from a dengue virus project to some initial work we've been exploring with the Aharoni lab at the Weizmann Institute about the possibility of applying these methods to plant metabolomics. So I've told you about a lot of work, of which I'm very grateful for help from many, many people. We've had fantastic collaborators here at MIT. I hope I've mentioned their names individually along the way, and collaborators around the world as well. And I've been privileged to have fantastic people in my lab, and I want to thank those of you who are here right now, and also past members of the lab who really contributed fantastically, and many have gone on to their own scientific careers. With that, I'll stop and be happy to take any questions.